I'm Emily Vlasnik, and for my STEM project, I did an analysis of mouthwash. I compared a name brand Listerine mouthwash to a generic mouthwash rinse. First, I analyzed the food dyes in each sample using a spectrophotometer. The graph on the left shows the Listerine mouthwash sample, and the graph on the right shows the blue 1 food dye. The peak is not relevant to this experiment, as that is the amount of the absorbance. The wavelength is what we look at, and since the peak occurs at the same wavelength, there's blue 1 food dye in the Listerine mouthwash, which is what is labeled on the bottle. I repeated the same process with the generic mouthwash. The generic graph on the left matches that of the green 3 food dye graph on the right, proving that there is green 3 food dye in the generic mouthwash, which is as it is labeled on the bottle. Next, I analyzed the amount of alcohol in each mouthwash sample. To do this, I first diluted each sample in 1,000 milliliters of distilled water. I then added 10 milliliters of acid dichromate to 6 flasks, 3 for each sample, and suspended 1 milliliter of the diluted sample above, sealing it with a rubber stopper. I then put all the samples into an incubator overnight to allow the alcohol to evaporate. I took it out the next day and let the alcohol sit and recondense. Then, rinsing the sides of the flask, I got all of the ethanol off of the sides and into the solution. I then added 1 milliliter of potassium iodide, which is an indicator, to get the brown color pictured above. I then began my titration by slowly adding sodium thiosulfate until the solution became a pale yellow, which took about 15 milliliters. This is showing that it is becoming more neutralized. To continue the neutralization, I then added 1 milliliter of the starch solution. This would neutralize the unreacted dichromate. I had to add this in small drops, one at a time, because the titration happened really quickly, and if you added one too many, it would have been too much. So you had to be very careful when it started changing and the blue started going away to stop adding sodium thiosulfate and just swirl it till it was clear. I then repeated the same process for Listerine. It took about 16.95 milliliters of sodium thiosulfate to neutralize the generic, and about 17.65 milliliters to neutralize Listerine. And this is a picture of the setup of the titration. Here is a time lapse of me performing the titration. I apologize, it does cut off at the end, but the picture in the bottom right hand corner will show the final product after I added the starch solution and it turned blue. I then added small drops, which is where the air comes in. If I added 1 drop too many or not enough, the results would be altered. But after you get it to a certain point, just swirl it and it will eventually become clear. I then repeated the same process to create blanks with just acid dichromate and no mouthwash sample, and therefore no alcohol. It took an average of 22.68 milliliters of sodium thiosulfate to neutralize this solution. And comparing it to the mouthwash samples and doing my calculations, I came to find that 15.8% alcohol was in the Listerine mouthwash and 14.0% was in the generic brand mouthwash. The bottle claims that there's 21.6% in each, which tells me that there can be some error in my lab results. Since my calculations were off by about 6 or 7%, I had to account for some error. Like I've said before, adding too much or not enough sodium thiosulfate can alter the results. Also, when I was diluting, I might not have added the same amount of each sample. My measurements might not have been exact. And when I was putting in 1 milliliter of mouthwash, I did have trouble with the pipette leaking, so that could have changed my different samples, which could have changed and affected my averages. That is the end of my presentation. That is the experiment that I did. I appreciate you taking the time to watch it. Thank you for listening.